Welcome to Department of Collegiate and Technical Education. This video is on Water Supply Engineering for the third semester Civil Engineering Diploma program. The objective of the course outcome is to ascertain the quality of water as per the Bureau of Indian Standards. So this is section number 18 in the unit 2 quality of the water. In the previous sessions, in the earlier sessions, we discussed about the impurities in the water. So, suspended impurities, colloidal impurities, and the dissolved impurities in the water. And what are the sources of impurities? And then we discussed about the examinations of water, that is the analysis of the water. So, physical characteristics of water are analyzed, like temperature, turbidity, odor, etc. Chemical properties, chemical characteristics of the water are found by measuring the hardness, chloride, fluoride, etc. So, in this session, we will continue the session by dissolved oxygen gases in the water, dissolved gases in the water, and the bacteriological characteristics of the water. So, dissolved gases. So, various gases are absorbed by the water when it is in contact with the atmosphere and also the ground surfaces. So, the gases that are absorbed by the oxygen are nitrogen, methane, hydrogen sulfide, carbon dioxide, oxygen, etc. So, in this, nitrogen is not much significance in water. Methane may be studied for its explosive characteristics. This hydrogen sulfide, even if it is found in small quantity, it gives odor, a very bad smell, and also a very bad taste to the water. This carbon dioxide gives smell and taste to the water, and it causes corrosive properties. This oxygen. So this oxygen is generally absorbed by the water from the atmosphere. From the atmosphere, the oxygen is absorbed. But this oxygen absorbed is consumed by the unstable organic matters and use it for oxidation processes. The complex organic substances are converted into the simpler substances by consuming the oxygen. Hence, the oxygen level indicates the amount of organic substances present in the sample. So, the extent of the organic matters may be studied by supplying the oxygen to the sample, and after consumption, by observing the amount of oxygen left out, we can easily calculate the amount of oxygen matter present in the water. So water dissolved oxygen. So water absorbs the oxygen from the atmosphere. This dissolved oxygen keeps the water fresh, sparkling, and it gives taste to the water. But this dissolved oxygen is consumed by the organic substances for their biological decomposition. So what is the amount of oxygen required for the biological decomposition, complete biological decomposition of the organic substances is measured by a parameter called biochemical oxygen demand. So as I already said, here what we do is oxygen is supplied to the sample, and after oxidation, so the amount of oxygen left out is noted, and it indicates the amount of oxygen consumed and the organic matter present in the water. So it is a measure of amount of oxygen demanded by the living organisms to oxidize the organic matter. The amount of oxygen required by the organisms to oxidize the organic matter is called the biochemical oxygen demand. So if sufficient oxygen is present in the water, this oxygen is consumed by the aerobic bacteria. So aerobic bacteria are the bacteria which are dependent on the oxygen for their survival. So if sufficient oxygen is present in the water, these useful bacteria, aerobic bacteria, cause the biological decomposition of the waste. They convert the waste into the simpler particles. They decompose the waste, organic waste, into simpler substances by utilizing the oxygen. So BOD is an indicator of the extent of organic matter present in the water. So actually we cannot wait until the complete oxidation occurs. Hence the BOD of the water during the first 5 days at 20 degrees Celsius is taken as the standard for the BOD. So BOD indicates the extent of the organic matter present in the water, and it also indicates the extent of treatment required. If we calculate the organic matter present in the water, the degree of treatment to be given in the purification plants is to be ascertained. So how this test is conducted? So the BOD test is conducted by diluting the sample with pure water of non-dissolved oxygen. So first what we do here is the sample of water is taken, then it is diluted with the water, pure water, with the non-dissolved oxygen content. So the dissolved oxygen content of this water, pure water, is known. That pure water is added or diluted with the raw water sample. That is that sample whose BOD is to be tested. So it is kept in an incubator. So incubator is a device which maintains the constant temperature. So this is incubated for 5 days at 20 degrees Celsius. So after 5 days, the oxygen in the sample is consumed by the organic matters. So what is the residual oxygen left is measured. So the difference between the original oxygen content and the residual oxygen content indicates the amount of oxygen consumed. So what I am saying here is, first the initial dissolved oxygen amount is known. So the sample is diluted with a water of non-dissolved oxygen. The initial dissolved oxygen is known. It is incubated for 5 days at 20 degrees Celsius. So if there is organic matters, so these organic matters consume the oxygen and it is utilized for the biological decomposition activities. So after consumption of the oxygen, so what is the oxygen that is left out? That is called the residual oxygen. This indicates the amount of the oxygen consumed. So the amount of the oxygen consumed indicates the amount of the biological activity or the amount of the organic matters. So this is an indication of the extent of the organic matter present in the water sample. That is called the biochemical oxygen demand. So what if BOD of water is 0? What it indicates? So there is no biochemical oxygen demand. So if no oxygen is required, means what? No organic matter is present. So there is no organic matter. So no oxygen is required to oxidize the organic matter. Hence, if BOD of water is 0, it indicates that no oxygen is required and no organic matter is present. So what should be the BOD of the treated water, or what should be the BOD of the water that is to be supplied to the public? So BOD for safe drinking water should be nil. So it should be nil. There should be no oxygen demand, or there should be no organic matter present in the sample. Now let us discuss about the bacteriological examination of the water. So what is bacteriological examinations? So first of all, let us understand what is bacteria. So bacteria are the minute single-celled organisms without definite nucleus. So these are the bacteria are the single-celled organisms with no definite nucleus. So most of the bacteria are not harmful to the humans. In fact, some of them are beneficial to the plants, animals, and humans also. So some of the bacteria are not harmful, and such bacteria are called non-pathogenic bacteria. So the bacteria which are not harmful are called the non-pathogenic bacteria. However, some of the bacteria are harmful and they spread the diseases, waterborne diseases like cholera, typhoid, dysentery, jaundice, etc. So such a type of bacteria which cause diseases are called pathogens. The disease causing bacteria are called the pathogens. ರೋಗಕಾರಕ ಬ್ಯಾಕ್ಟೀರಿಯಾಗಳು ಅಂತ ಕರಿತೀವಿ. So such type of bacteria are called pathogenic bacteria. So pathogenic bacteria are the bacteria, disease causing bacteria, and non-pathogenic bacteria are the bacteria which are not harmful. Now most of the bacteria require oxygen for their survival. So they consume the dissolved oxygen from the water and decompose the organic matters present in the water. They convert the organic matter into the simpler substances. So these aerobic bacteria present, they require oxygen for their survival. So they act upon the organic materials, organic compounds present in the water, so decayed vegetation, etc. So they convert this organic matter into simpler substances. So these type of bacteria which requires oxygen for their survival are called aerobic bacteria. Some of the bacteria does not require oxygen for their survival, and such bacteria are called the anaerobic bacteria. So bacteria, some of them are harmful, some of them are not harmful. The bacteria which requires oxygen for their survival are the aerobic bacteria, and that bacteria which do not require oxygen for their survival are anaerobic bacteria. So why this bacterial analysis is to be made? So bacterial analysis is a method of analyzing the water to estimate the number and form of the bacteria present in the water. So this process is used to check whether the water is safe for the human consumption. So for a safe drinking water, there should be no pathogens present in the water. So bacteria are the single cell organisms with no definite nucleus. Most of the bacteria are harmless and they are called non-pathogenic bacteria. Disease causing bacteria are called the pathogenic bacteria. Pathogenic bacteria causes waterborne diseases. The bacteria which requires oxygen for their survival are called the aerobic bacteria. The bacteria which does not require oxygen for their survival are called the anaerobic bacteria. So what are the bacteriological tests that are conducted on the water? So bacterial tests conducted on the water are, first one is the total count test. Second one is the B. coli test. So what are these tests and how these tests are performed and what is the significance of these tests? Let us study here. So total count test, as the name indicates, we count the number of colonies of bacteria in the water. So what we do here is, so the sample is taken, the water samples are collected and they are placed in the different dilutions of water. That is, the samples are placed in different dilutions, that is a set containing the 10 ml of sample, a set containing 0.1 ml of the sample, and a set containing 100 ml of the sample. Like that, different sets of the samples are taken. A set containing 10 ml of the sample, a set containing 1 ml of the sample, and a set containing 100 ml of the sample. Like this, different sets of the samples are taken. To it, a culture media is added. Culture media of agar is added. So what is this culture media? So this culture media is the nutrient for the bacteria. That is, they grow by these nutrients. So some environment is created for the culture of the bacteria. So some nutrients are added for the bacterial growth. So this culture medium is provided in all the samples collected. So we have prepared the samples in different dilutions, and we have placed the cultured medium of agar in all the solutions. So these medias are incubated in they are incubated for 24 hours at 37 degrees centigrade. So these samples are placed in the incubator for 37 degrees centigrade for the 24 hours. One day it is kept in the incubators at 37 degrees centigrade. So we have taken the samples, we have put the culture media in it, and we are maintaining the temperature of 37 degrees centigrade for 24 hours. These are the favorable environment for the growth of the bacteria. So after 24 hours, the bacterial colonies formed are counted. So by using the microscope, we observe the number of bacterial colonies formed in the samples, different sets of samples. So if the bacteria are the number of bacteria, the total count of the bacteria is taken here. So this is the total count test. So what we have done here is, we have taken as different sets of samples, we have placed a culture media in it, then this is incubated for one day at 37 degrees Celsius. So during this, if there is any bacteria colony are observed, that their total count is taken. That is the total count test. The second test is the B. coli test. So B. coli or coliforms are the rod-shaped non-pathogenic bacteria whose presence or absence indicates the pathogens. So B. coli test, we are doing the B. coli test. So these coliform bacteria are non-pathogenic, that is, they do not cause any diseases, they are not harmful. These bacteria are taken as an indicator for the presence of the pathogenic bacteria. So what I am going to say is, so testing the bacteria which are pathogens, that is disease causing bacteria, is difficult. So the presence of the pathogens in water is difficult to find. So instead of this, what we do is, the coliform bacteria are the non-pathogenic bacteria which are not harmful, and we find the presence or absence of the coliform bacteria. It also indicates the presence or absence of the pathogens. We assume that they originate from the same type of culture. So coliform bacteria, the presence of the coliform bacteria indicates the presence or the absence of the pathogens. So another principle is, this B. coli bacteria are lactose fermenters. That means to say that if they convert they ferment or they convert the lactose into acid and the gas, lactose materials into acid and the gas. So if lactose environment is there, they convert the lactose into acid and gas. So if B. coli bacteria are present, this lactose is converted into the gas. So this principle is used in counting the bacteria. So B. coli test involves 3 tests. So first one is the presumptive test, second one is the confirmed test, third one is the completed test. The test on the B. coli test involves 3 steps. First one is the presumptive test, confirmed test and completed test. Let us see what are these presumptive, confirmed and completed tests. So what we do here is, first we water sample is taken and a presumptive test is done on the water sample for finding the B. coli bacteria. A test is conducted in the by presumptive test. So this test is conducted, and if positive result comes, that is if B. coli bacteria are present in the presumptive test, then this sample is taken for the second test, that is confirmed test. If the negative results comes in the presumptive test, that is if the negative results, that is no bacteria or no B. coli is present in the sample. So if the in the presumptive test, if there is a positive result, so it is taken into the second step, that is confirmed test. In the confirmed test, the sample from the presumptive test is taken, and here again test is conducted. If negative results comes, it indicates that no B. coli bacteria are present. If positive results comes, it is for if bacteria are observed, then it is again a completed test is conducted. So completed test is conducted to confirm the presence of the bacteria. So I hope you understand the flow diagram. So how this is done? So presumptive test. So in the presumptive test, what is the sample we are going to take? So the sample whose bacterial analysis is to be done, that sample is taken, that is the diluted sample of water is taken. So here agar is used as the culture media and lactose broth is added to the solution. So we know that B. coli bacteria ferment or convert the lactose broth into acids and gases. If bacteria are present, this lactose broth is converted into acids and gases. If no bacteria are present, this gas is not produced. That's a simple thing. If bacteria are present, gas formation is formed. This lactose is converted into acid and gas. If no bacteria are present, so no gas is formed. So this diluted sample of water is taken and lactose broth is added. This is maintained at a temperature of 37 degrees centigrade for 48 hours. If there is a formation of the gas is formed, noted down. If there is a formation of the gas, it indicates that the B. coli bacteria are present. So hence this is unsafe for the drinking. Positive result indicates that the gas is formed and it is unsafe for the drinking. So what we have to do? So we are getting a positive result. So bacteria are present. So we go for the second test. That is the we go for the second test. If negative result comes, what happens? The sample is free from the B. coli. So no bacteria are present in the sample and it is safer. So that is the presumptive test. So observe this. Here the diluted sample is taken, lactose broth is added, temperature is maintained for 37 degrees centigrade for 48 hours. So if gas formation is noted, it indicates that bacteria are present, so unsafe for the drinking. So if negative result comes, sample is free from B. coli bacteria. So then we go for the confirmed test. So here the sample from the presumptive test is taken. Sample we have tested for the presumptive test, so that sample is taken. Here the culture media added is EMB agar. So EMB agar culture medium is added to the sample. The condition of the test is 37 degrees centigrade is maintained for 24 hours. If colonies of bacteria are seen under the microscope, then it indicates that it is a positive result. After 24 hours, if colonies of bacteria are observed, it indicates that it is a confirmed test. Negative result, if the sample is free from the E. coli bacteria, that is a negative indication. So if bacterial colonies are observed in the confirmed test also, then what we have to do? We go for the completed test. So here we take the sample from the confirmed test. We take the sample from the confirmed test. Here lactose broth and agar tubes both are taken. So the condition of the test is here 37 degree temperature is maintained for 24 to 48 hours. If gas is formed, positive result is confirmed and the sample is not safe for drinking purposes. It is contaminated with the bacteria. If negative results comes, the sample is free from the E. coli bacteria. So this is the complete picture of the E. coli tests on water. Hope you understood. So E. coli index. So this is the index number showing the approximate number of E. coli bacteria per sample of water. So for a sample of water, this is an indication of how many approximate number of E. coli bacteria are present. This is called the E. coli index. For a potable water, that is for the drinking water, drinking purpose, the E. coli should be between the 3 and 10. Most probable number, MPN. So it is a statistical estimate of the number of coliform group organisms per unit volume of the sample of water is called the most probable number. Here what we do is, in the previous test we have taken samples of different dilutions, 0.1 ml dilutions, 1 ml dilutions, 10 ml dilutions. So we have taken different samples. So the result of each sample is noted as positive and negative, and statistical estimate, a probable estimate of the bacteria present may be worked out by using the statistical estimates, and that is called the most probable number. So it is expressed as density of the coliform per 100 ml of the sample. So number of bacteria present per 100 ml of the sample is taken as the MPN index. Now let us talk about waterborne diseases. So what are the waterborne diseases? So the diseases caused by the bacterial infections are thyroid, cholera, dysentery. So these are the diseases caused by bacteria. What are the diseases caused by viral infections? Infectious hepatitis, that is jaundice. Diseases caused by protozoa infections are amoebiasis or amoeboic dysentery. So waterborne diseases are, we have to specially care for the waterborne diseases to spread the epidemics. So special care has to be taken in case the epidemics persists. So waterborne diseases can be prevented by observing the following points. So before we supply the water to the public, proper treatment has to be given to the water. We have to supply always the wholesome water, that is the water which is free from the objectionable impurities. That water has to be supplied to the public. So not laying water supply line and sewer lines nearer. So sewer lines and water supply lines should not be laid nearer to each other, otherwise there will be mixing or contamination takes place. Proper checking of the water supply lines has to be done for any leakages of water or any contaminations. We have to observe periodically. So sealing of the leakage points. If there is any leaking points are there, we have to seal it properly. So we have to drink boiling water. ಸೊ ಬಿಸಿ ನೀರು ಕುಡಿಯೋದು ಒಳ್ಳೆಯದು ಅಂತ ನಾವು ಹಿಂದಿನಿಂದ ಹೇಳ್ಕೊಂಡು ಬಂದಿದ್ದೀವಿ. ಸೊ ಬಿಫೋರ್ ಕನ್ಸಂಪ್ಷನ್ ವಾಟರ್ ಇಸ್ ಟು ಬಿ ಬಾಯ್ಲ್ಡ್. ಸೊ ಇನ್ಕ್ರೀಸಿಂಗ್ ದ chlorine dosage. So in case of epidemics, more chlorine is added for disinfection purposes. Maintaining the city clean. So maintaining the city clean stops most of the epidemics. So this is about the bacteriological investigations of the water. So let us solve some multiple choice questions. So can you name any two dissolved gases in water? So what is the answer? Carbon dioxide, hydrogen sulfide, oxygen. These are the dissolved gases in water. Bacteria requiring oxygen for the survival are called what? So bacteria which are dependent on the oxygen for the survival are called what? Aerobic bacteria, anaerobic bacteria, both, none of the above. So the bacteria requiring oxygen for survival are called the aerobic bacteria. Name the two tests on bacteria. So what are the tests that are conducted for bacterial analysis? Total count test and B. coli test. State true or false. BOD is an indication of organic substances in the water. So biochemical oxygen demand indicates the organic substances present in the water. State true or false. Yes, organic substances requires oxygen for oxidation. So BOD is a measure of oxygen consumed. State true or false. BOD test is conducted for 5 days at 20° temperature. Is it true? Yes, BOD is conducted for 5 days at 20° Celsius. I hope you understood the concept. Thank you.